Hello, hi, a very happy good evening. So this is Krishnaveni and we've been talking about the story of life on earth, and that's biology. So welcome to my class. Today we are in lecture 3 of Cell, the unit of life. Hi Nandini, after a very long time you are here. Yes, I am very happy good evening. I'm good, thank you. How are you? So thank you so much for joining my lecture today. So please do like, share and subscribe. So lecture 3, Cell, the unit of life. So we were talking about your eukaryotic cells. So today we'll talk about your endomembrane system of your eukaryotes. Okay. So before that, so please do like, share and subscribe. So let your friends know that here I am teaching the whole of biology, class 11, class 12, botany, zoo, everything I teach. So tell them. And we're also discussing questions. We are also having Guess the Series mentee. Okay, so please share the link with your friends. So shall we talk about Cell, the unit of life? Yes. So it's lecture 3. So before we go into your endomembrane system, I have got some questions. So let's solve the questions as well. Yes, shall we move into the lecture? So before we go into today's lecture, so I have an announcement for tomorrow. So tomorrow at 4 p.m. we are discussing important questions from the topic respiration in plants, right? So we have not yet completed, so one small portion is remaining. So we'll complete that, and tomorrow we'll discuss important question in respiration in plants. Okay, and at 6:30 p.m. so again we have guessed the chapter. So it is episode 4 tomorrow. So please join me for the same. Hi Vairavan. Yes, a very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. So this is the schedule for tomorrow. So please make a note of it and join me for the same. Okay. Yes. So lecture 3 of Cell, the unit of life. So what were we discussing in this topic, Cell, the unit of life? So we were we discussed what is a cell. So cell is what makes us feel living, right? So that is how we differentiate ourselves from the non-living thing. Then who gave the cell theory? Overview of the cell. Then prokaryotic cell, and finally eukaryotic cell. So hi Munna. So today Munna is before than Shimpi. Okay. So Munna, yes, hello, hi. Thank you so much for joining my session. I'm good, thank you. How are you? So Shimpi, hello, hi. Yes, a very happy good evening. I'm fine. How are you? So please do like, share and subscribe. It's raining in Kota right now. Okay. So let's talk about what I discussed in the first 2 lectures. I will give you a quick recap of the first 2 lectures. So we started talking about the cell. So how the cell acts as a superhero in biology. So then we went on to talk about the cell theory. So the cell theory was given by Schleiden and Schwann. But they failed to say where the cell comes from. So the cell comes from the pre-existing cell that was given by Virchow. Then he modified the cell theory. So all products, so all organisms are made up of products of cell, and a cell comes from pre-existing cell. So these are the postulates that he gave. So then we went on to talk about the difference between a prokaryotic and a eukaryotic cell. So you guys told me quite a lot of differences. So prokaryote and eukaryote. So both the prokaryote and eukaryote, they have a semi-fluid matrix. So that is known as your cytoplasm. So this is present. So your eukaryotes have membrane-bound organelles. And they have well-defined nucleus. That is, the nucleus is surrounded by a nuclear membrane. So in case of prokaryotes, they do not have any membrane-bound organelle. They only have non-membrane bound ribosomes present. So ribosomes are essential for protein synthesis. Okay, so now only you watched my previous 2 lectures. So thank you so much, Vairavan. So hi Satyendra, hi Anthony Abhi. So thank you so much for joining my lecture. So please do like, share and subscribe. Okay, so this is about the overview of the cell. And your animal cells, so prokaryote, eukaryote are different. Animal cell, plant cells are different. So both your animal cell and your plant cell comes under your eukaryotes. Okay. So Nari Jan, hello. Yes, thank you so much for joining my lecture. I guess you're new to my lecture. So we are talking about cell, the unit of life. So this is lecture 3. So I'm giving a recap of the first 2 lectures. So if you have any doubts, so stop me there and ask me. Okay. Yes. So animal cell and plant cell both are eukaryotes. Hi Keshav, what a surprise, you're here for the next lecture also. So you have a school class at 7? Yes. Okay, anyways, thank you so much, Keshav, for joining my lecture. So please do like, share and subscribe. So animal cells have no cell wall. So plant cells have cell wall. So further, animal cells have another non-membrane bound structure that is known as your centromere. So plants do not have. So they have what is known as your vacuoles. Right, so this is the difference. So first we talked about your prokaryotic cell. So in your prokaryotic cell, what did we discuss? So all your prokaryotes except your mycoplasma, they have cell wall. So all prokaryotes except mycoplasma, they have your cell wall. Right, and how is the cell membrane? Yes, plastids, that is also an essential point. Yes, Vairavan, you're right. So then they have a cell envelope. So how is the order? So they have glycocalyx, then you have the cell wall, then you have the plasma membrane, right? So your glycocalyx can take two forms. It is a slime sheath when it is loosely arranged, and it is a tough capsule when it is tightly packed, right? So this is the order. So based on this, they can be divided into two types, that is gram positive and gram negative bacteria. So gram positive bacteria has a thick layer of your peptidoglycan. Yes, after that they have your lipid bilayer. So here you have a lipid layer, you have peptidoglycan, and again you have your lipid. So this is the difference between gram positive and gram negative. So hello Surender, yes, a very happy good evening. Wow, after a very long time, it's really good to see you. So thank you so much for joining my lecture. So please do like, share and subscribe. Okay. Negative is sandwiched model, okay, because it has a peptidoglycan layer which is sandwiched between the lipid bilayer. Your gram positive, so why it is called as gram? Because it was discovered by Hans Christian Gram. So he was the scientist who found that. And why positive? Because they are able to retain the strain or the dye because of the thick peptidoglycan layer. So it is a polymer, right? So your gram negative is not able to retain the dye or the stain because of the thin peptidoglycan layer. So your gram positive are pathogenic, but your gram positive are not pathogenic. So what do I mean by pathogenic? They are disease causing. So your gram negative are pathogenic. So we saw about this. Then we went on to talk about your ribosomes. So ribosomes have two subunits. So you have a larger subunit that is 50S, a smaller subunit that is 30S. So what do they need for joining? So they need your Mg2 plus ions, right? So together when they are joined, they make up 80S ribosomes. So ribosomes are needed for the translation of proteins. This is the third part of your central dogma. Okay, so E. coli is gram positive or gram negative. So E. coli is gram negative. It is not that all gram negative are pathogenic. Okay, some are good, but majority are gram negative. They cause diseases. Okay, yes, fine. So this is about your ribosomes. Then we started talking about your inclusion bodies. So inclusion bodies, they store your reserve food material. So you have your sulfate granules, cyanophation granules, phosphate granules, etc. So they all come under your inclusion bodies. Fine. So then we went on to talk about your eukaryotes. So in eukaryotes, we spoke about a lot of things. So we started off with the cell wall in eukaryotes. So cell wall is present in plants, right? So cyanobacteria is gram negative. Yes, you're right. Okay, fine. So in eukaryotes, the cell wall is present only in case of plants. So in plants, you have a middle lamella layer which contains calcium pectate molecules which helps in cementing neighboring cells together, right? So then what else did we talk about in eukaryotes? Yes. So then we started talking about the endomembrane system. So no, sorry, we started talking about the plasma membrane, the quasi nature of the plasma membrane enables the movement of molecules. Then we spoke about the lipid bilayer. You have a polar head and a non-polar tail, right? So this is how your lipid bilayer is arranged. So cyanobacteria, is it disease causing? To some extent. Not all cyanobacteria are disease causing, but some of them are disease causing, okay? Singer and Nicholson gave the quasi-fluid model. Yes, you're absolutely right. So in the next class, I will get you a list of gram-negative and gram-positive bacteria. So looking at the bacteria, you can know which causes infection, which does not cause infection. Okay? Yes? Fine. So this is what we read in the first two lectures. So based on this, I have some questions. See if you can answer them. So question number 1, an organelle which is devoid of a membrane covering is. An organelle devoid of membrane covering is vacuole, ribosome, peroxisome, lysosome. Yes, what is the answer? Come on, make it quick. Yes, what is the answer? So ribosome, others, Anthony, Abhi, Shimpi, Keshavan, what is the answer? An organelle devoid of membrane covering? Yes, all of you accept it is ribosome. So vacuole has a membrane, that is it has a membrane, that is tonoplast. Peroxisome is in case of plants. The plants who undergo C2 pathway, they contain peroxisomes. Lysosomes also have a coat, also have a membrane, okay? Yes, DJ Films and Entertainment, thank you so much for your support. So please do like, share and subscribe. So this is about your question 1. So moving forward, question 2, the difference between prokaryotic and eukaryotic cell is in having cell wall, nuclear membrane, ribosome, none of these. What is the answer? So the difference between the prokaryotic and the eukaryotic cells in having the cell wall, the nuclear membrane, ribosome, none of this. Yes, Keshav. So the major difference between prokaryotic and eukaryotic is the nuclear membrane. The nucleus is not enclosed by a nuclear membrane. Yes, absolutely right. Okay, good. So question 3. So Shimpi right, Vairavan good. Satyendra also gave me the right answer. So question 3, extension of the plasma membrane in prokaryotic cell is mesosome, haploid, ribosome, none of these. What is the answer? Extension of the plasma membrane in prokaryotic cell is your mesosome, is haploid, is ribosome, or none of these. Come on, make it quick. Yes, what is the answer? Extension of plasma membrane in prokaryotes is? Yes, perfect, Shimpi. They are mesosomes, the infolding of the plasma membranes. So you have infoldings. They are found to contain your respiratory enzymes. So they mimic your mitochondria in your eukaryotes. Respiratory enzymes, right? So they increase the surface area. So it is your mesosomes, right? So question 4, polysome is a chain of, yes, Nari, you're also right. Okay, so everyone has given me the answer. So polysome is a chain of oxysomes, spirosomes, ribosomes, dictyosomes. So polysome is a chain of oxysomes, spirosomes, ribosomes, dictyosomes. 4C, ribosomes, yes, absolutely right, Keshav. So polysome is when to an mRNA, if your ribosome goes and binds, so this is known as your polysome, right? So it is option C. What are spirosomes? What are dictyosomes? I will discuss in the last topic, that is your microbodies. There I will give you the description. Okay, so here it is option C. So question 5, integral proteins of the cell membrane occur on in the inner surface, outer surface, phospholipid matrix, inner and outer surface. Yes, what is the answer? Integral proteins of the cell membrane occur on in the inner surface, the outer surface, phospholipid matrix, inner and outer surface. Phospholipid matrix, so matrix is the center portion. Matrix is the center portion. Yes, Satyendra, you're right, the inner and the outer surface. So this is my phospholipid bilayer. Right, so it is present like this. So the proteins that are present here are my peripheral proteins. So the proteins that are present like this, they are my integral proteins. So it is present on the inner as well as on the outer side. Yes, Nari, you're right. So it is option D. Okay. So question 6, the active transport across biomembrane involves. The active transport across biomembrane involves the production of ATP, requirement of energy, production of toxin, release of energy. Yes, what is the answer? So active transport across biomembrane involves production of ATP, requirement of energy, production of toxin, release of energy. Yes, active transport requires energy. Absolutely right. So the membrane of erythrocytes has approximately dash of proteins and dash of lipids. So this is a direct statement from your NCERT. Yes, what is the answer? What is the percentage of proteins and what is the percentage of lipids in your RBC? So in the membrane, in the plasma membrane of your RBC, yes, what is the percent? Quick. Yes, absolutely right. So it's 52% proteins. And it's 40% lipids. So option B is the right answer. Okay, so question 8. The lipid components of the membrane consist of polysaccharides, phosphoglycerides, monosaccharides, both A and C. The lipid component of the membrane contains what? Mainly consist of polysaccharides, phosphoglycerides, monosaccharides. Yes, what is the right answer? Yes, good. All of you have given me the right answer here. Okay, very good. Yes. Tell me. The lipid component of the membrane mainly consists of polysaccharides, phosphoglycerides, monosaccharides, both A and B. So Shimpi says A. Why only polysaccharides? The lipid components. Polysaccharides are your carbohydrates. So the lipid component of the membrane consists of your phosphoglycerides, right? So it is option B, it is phosphoglyceride. No, Satyendra, it is phosphoglycerides. Okay. So moving forward to the next question. So question number 9. In which of the following the cells are held together by a calcium pectate layer? Primary cell wall, secondary cell wall, middle lamella, tertiary cell wall. Come on, it's an easy question. It's an easy question. Calcium pectate layer is present where? Quick. Yes. Yes, it is an easy question. So calcium pectate is present in your middle lamella. Right? Everyone agrees? Yes, Vairavan is right. Satyendra is right. Yes, Nari, you're also right. Yes, Shimpi, perfect. So the last question. So which one of the following structures between two adjacent cells is an effective transport pathway? Which one of the following structures between two adjacent cells is an effective transport pathway? Plasmodesmata, plastoquinones, endoplasmic reticulum, plasmolemma. So Falguni Sharma, hello, hi. So I guess you're new to my session. So welcome to my session. So thank you so much for joining. So please do like, share and subscribe. Yes, what is the right answer? Yes, what is the right answer? Yes, perfect. So plasmodesmatal connections are like this. They are present between the cells. So this is your plasmodesmatal connections, right? So the right answer is plasmodesmata. So whatever I have taught before, is it clear? So hi Naveen, how are you? After a pretty long time you are here. So please do like, share and subscribe. So we are discussing cell, the unit of life. So this is lecture 3. So I have given some questions for lecture 1 and lecture 2. So we were discussing them. So no, Falguni Sharma, it is not plasmolemma. So it is plasmodesmata, the connection between the cytoplasm of the adjacent cells. Okay. Yes sir, a very, very happy good evening. So welcome to my class. So please do like, share and subscribe. Yes, you're right, Vairavan. Yes, everyone is right. Ma'am, lipids are monosaccharides? No. You say monosaccharides only for carbohydrates. So you cannot say lipids as monosaccharides. So lipids are made up of your fatty acids and your glycerols. Yes, fine, clear. So shall we move forward? Okay, so with this, we are done with the introduction of your eukaryotic cell. So now let's talk about your endomembrane system. So here, when I say a system, it means 2 or 3 members are coordinated. A system is when you have a team. If I say computer is a system, there are so many parts which are integrated, which are coordinated, right? So much in the same way, when I say endomembrane system, you have members which coordinate to give you a specific function. So this includes your endoplasmic reticulum. So the first member of this is your endoplasmic reticulum. You can say for a team, you have a leader, you have a coordinator, and then you have team members, right? So much in the same way, endoplasmic reticulum is the leader. Okay, so it goes in this order. So from your endoplasmic reticulum, you have Golgi complex, then you have your lysosomes, and then you have your vacuoles. It moves in this order. So this is your endomembrane system. So a system is where you have members and whose functions are coordinated to give a particular output. So who are the members? So go in the order: endomembrane system, Golgi complex, lysosomes and your vacuoles, right? So moving further. So we were talking about your endoplasmic reticulum. So I stopped right here in the previous class, but let me start from here. So the network or the reticulum of tiny tubular structures that are scattered in the cytoplasm of the cell. So reticulum or network, right? So we have read about your reticulate venation. So it is a network, it has a lot of connection. So this is your endoplasmic reticulum. Ma'am, for both plants and animals, same order. So animals do not have vacuoles. In animals, the vacuoles are not well developed. But yes, the order is the same. Okay, Vairavan? Yes? So a network or reticulum of tiny tubular structures that are scattered in the cytoplasm. So the endoplasmic reticulum divides the intracellular space. So intra means within the cell. So ER divides the intracellular space into 2 distinct compartments. So within the cell you have your endoplasmic reticulum, right? So this divides it into 2 compartments, one inside of the ER that is known as the luminal side, and one extraluminal that is towards the cytoplasm. So inside your cell, in one corner you have your endoplasmic reticulum pleasant. So this is my luminal side. That is the inner side of your ER, and this is the cytoplasmic side. This is the extraluminal. So what divides? The ER divides the intracellular space into 2 distinct compartments. So ER, the ribosomes are attached to the surface like this. So this gives it a rough appearance. So it is known as rough endoplasmic reticulum. Okay. So absence of ribosomes gives it a very smooth appearance. So it is known as smooth endoplasmic reticulum, right? So what is the function of your rough endoplasmic reticulum? Protein synthesis and secretion. So whenever your ribosomes are involved, so remember the main function is protein synthesis. Okay, so what is the function of your smooth endoplasmic reticulum? It is a site of synthesis of lipids and your steroidal hormones. So I showed you this picture. So here you have ribosomes attached. So it is your rough endoplasmic reticulum. Where there are no ribosomes attached, this is your smooth endoplasmic reticulum. So this is towards the inside of your endoplasmic reticulum. So this is the luminal side. And this is the extracytoplasmic side. So your endoplasmic reticulum is clear? No doubts with your endoplasmic reticulum? Yes? Detoxification? Yes, you're right, but we will talk about it. Fine. So endoplasmic reticulum is present in all eukaryotes and it is absent in prokaryotes because prokaryotes do not have any membrane bound vacuoles, right? So it has none of them. So it is present in all eukaryotes except your germinal cells, that is your gametes, and your mature mammalian erythrocytes. Your mature mammalian erythrocytes also do not have nucleus and it has no cellular components. What is the composition of endoplasmic reticulum? It is made up of foldings, okay? So it is a network which is made up of foldings. So let's see what is the function of endoplasmic reticulum. So it is a system of membranes. It is composed of 3 types of forms. So this is the composition Falguni. So it consists of cisternae, vesicles and tubules. So point number 1, it is present in all eukaryotes except your germinal cells and your mature mammalian RBCs. So please remember the exceptions. So that is where everyone's eyes will go to. So please remember where it is absent. Yes, it is present in all eukaryotes, but it is absent in your germinal cells and your mature RBCN cells. It is present in immature cells, but your mature cell loses your nucleus, right? So thus it loses all your organelles. So Priya sweetie, yes, good evening, you're late to class, it's okay, we have just started your endomembrane system. So we are discussing about your endoplasmic reticulum. Thank you so much Priya sweetie for joining my lecture. So please do like, share and subscribe. Okay, so it is composed of 3 types. What is it? Cisternae, vesicles and your tubules. So what are cisternae? Right? So these foldings are known as your cisternae. Right? So vesicles are a small bag-like structure. So they are your vesicles and your tubules are tubular structures like this. I guess I have a picture. Okay, I don't have a picture. So your cisternae are foldings like this, right? And your vesicles are small sac-like structures and tubules are like this. So this is what, okay. So these are foldings, right? So each of them is known as a tubule and they form a cisternae. So sometimes they have sac-like structures known as vesicles. So this is the composition of your endoplasmic reticulum. Ma'am, NEET application is going to start today. Yes, even I saw the news like that, but I wanted to cross check it once, so that's why I didn't open my mouth about that. Okay, so I will confirm it tomorrow as to how and what you should do. So I will tell you tomorrow in class. Okay, yes. So smooth and rough endoplasmic reticulum, so which means your exam is scheduled on the 1st of August. So today is 2nd of June. So you just have 28 days and 1 month left, right? So almost you can say 58 days or 60 days exactly for your NEET exam. So please have some seriousness. So please prepare, solve questions. If you have doubts, get back to me, ask me. I will let you know. So because whatever you ask, I am replying, right? So even if it's not from the chapter I am teaching, I do get back. So please kindly be more serious. So it is months of hard work. Don't let it go in vain. So all of you have to come out with flying colors. So please pay some attention. So smooth endoplasmic reticulum and rough endoplasmic reticulum. So what is the main difference? So your smooth ER, the ribosomes are absent. So that is why it has a smooth surface, right? Yes, I will tell you tomorrow in class at 4 p.m. So because I want to cross check and I will go through the official website. Yes, so that is the reason I didn't open my mouth about it. So tonight I will go through it and tomorrow I will let you know in class. Okay? Yes. So smooth endoplasmic reticulum, it is smooth because it has no ribosomes. Rough endoplasmic reticulum is rough because of the ribosomes. Sorry, and your smooth endoplasmic reticulum is present near the cell membrane, that is towards the cytoplasmic side, and this is present near the nucleus, so this is towards the luminal side. Okay. How should be a 1-year NEET preparation schedule, ma'am, for NEET 2022? Okay. for NEET 2022, you have ample time. So read the NCERT, go chapter wise. So if you're following YouTube classes, so I have taught all the chapters in detail. So find out the links. So read the chapter, get yourself thorough in the concept, solve at least 150 to 250 questions from each chapter. So revise the chapter. So like this, finish off class 11, finish off class 12, and then go for full revision. So on an overall value, so on an overall scale, so this is how the preparation chart should be. Okay, Dharani? Yes. So your smooth endoplasmic reticulum is involved in the synthesis of fat, glycogen and steroids. So your rough endoplasmic reticulum is involved in the synthesis of proteins. So your smooth endoplasmic reticulum is only formed of tubules, but this is formed of cisternae and vesicle. So now do you understand? So your endoplasmic reticulum is made up of cisternae, vesicle and tubules. So your smooth one is only made up of tubules, and this is made up of cisternae and vesicles. See, so this has tubules like structure, right? So these are tubules, so these are cisternae, and you have vesicle like structures. The openings that are present, that are sac like structures, they are known as vesicles. Now clear? Yes, so your smooth endoplasmic reticulum is less stable, whereas your rough endoplasmic reticulum is more stable. So your smooth endoplasmic reticulum, examples are your adipose tissue because it is involved in fat synthesis, and your muscles. This is involved in protein, so your liver and your pancreas, right? So after becoming doctor, I will meet you. Yes, I am eagerly waiting to meet all of you as doctors. Okay. Yes, so this is the main difference. So moving further, what is the function of this endoplasmic reticulum? Yes, I know it is for protein production in case of rough endoplasmic reticulum, and steroidal production or fat production in case of a smooth endoplasmic reticulum. But apart from that, does it have any function? Yes, it has. So it acts as cytoskeleton. It provides mechanical support and shape. Cytoskeleton, so cyto means the cell. Okay, it acts as a cytoskeleton. It provides mechanical support and shape. It helps in the formation of primary lysosome, Golgi body and microbodies. So that's why I said endoplasmic reticulum is the leader of the endomembrane system, because it helps in the formation of lysosome, your Golgi body and your microbodies. Okay, so synthesis of nuclear envelope. So the main difference between prokaryotes and eukaryotes is that the nucleus is surrounded by a nuclear envelope. So this nuclear envelope is synthesized by your endoplasmic reticulum in the telophase of your mitosis. Your mitosis has prophase, metaphase, anaphase and telophase. So in the telophase, your nuclear envelope is synthesized by your endoplasmic reticulum. Then synthesis of lipids because of the smooth endoplasmic reticulum. Then synthesis of steroidal hormones again by smooth endoplasmic reticulum. So cellular metabolism, so these are the functions of your endoplasmic reticulum. So it acts as a cytoskeleton, it provides mechanical shape and support, it helps in the formation of your primary lysosome, your Golgi body and your microbodies, and it helps in the formation of your nuclear envelope in your telophase, synthesis of lipids, synthesis of steroidal hormones and your cellular metabolism. Done? Yes. So now the next member, that is your Golgi apparatus. So here we talk about your Golgi apparatus. So why is it named as Golgi? Because a scientist named as Camillo Golgi discovered it. He was the first to observe the densely stained, again a reticular structure near the nucleus. So once again it is a reticular structure, meaning it is a network of structures. So this consists of flat dish-shaped sacs or cisternae. So there also you had cisternae. Here also you are having cisternae, but these are flat dish-shaped structures. So this is known as your cisternae. Done? So concentrically arranged near the nucleus. So what do I mean by concentric? So these circles are concentric, meaning they have a center. So if nucleus is arranged, if nucleus is present, they are concentric around the nucleus. Okay, and they have two sides, convex or cyst forming phase and convex or trans-maturing phase. Just like your endoplasmic reticulum had a luminol phase and a cytoplasmic phase. So this has two phases, convex and concave phase. Okay, so concave is cyst forming and convex is cyst forming, concave is transforming. Okay, right? So look here, so this is cyst facing, so meaning this is convex, so convex like this. So this is concave, so this is trans facing or concave facing. So this has flat dish shaped structures, so these are known as your cisternae. Clear? Yes, so you are able to understand the structure of your Golgi body. So see how it is present. It is present around the nucleus. So this is concentric to the nucleus. So around the nucleus you have the endoplasmic reticulum and here you have your Golgi apparatus. So they have flat disc shaped structures known as cisternae. So cisternae comes in case of your endoplasmic reticulum, your Golgi apparatus. You have another term known as cristae. Cristernae or cristae. So this comes in case of your mitochondria. So don't get confused. So cisternae comes in case of your endoplasmic reticulum and your Golgi apparatus. So what is the function of Golgi apparatus? Why should we have a Golgi apparatus? So packaging materials to be delivered either to the intracellular targets or secreted outside the cell. So if you are going to courier something, right? So don't you pack it properly? It has to travel some distance, so you have to pack it and send it, right? So if you are ordering something in your Amazon platform, so how does it come? It's not that if you're ordering a book, they open their link, click on your doorbell and just give the book. No, it is packed. Your address is pasted on it so that it's known where it has to be dispatched. Similarly, your endoplasmic reticulum produces proteins, right? So your Golgi apparatus packs these proteins. For example, if you go to the post office, you should have seen letters spread around. So this is for this pin code, this is here, this is here, this is here. So similarly, your Golgi apparatus helps in packaging. It packs the proteins and based on the address it separates them. So this is inside the cell, this has to go outside the cell. It separates them like this. So that is the role of your Golgi apparatus. So Golgi body receives materials from your endoplasmic reticulum, that is the cis phase. So the cis phase is the convex phase, right? So this is close to your endoplasmic reticulum. So endoplasmic reticulum receives proteins and it produces proteins, it enters the Golgi apparatus, it is packed and it moves out through the trans phase. Okay? Yes? Lot of ordering in Amazon platform, so which means you're ordering a lot of things. Huh? So materials are chemically modified by the Golgi body. So if I suppose I'm a postman and I have 100 parcels. So it has to be delivered in so many states in India. So depending on the way it has to travel and depending on the size of parcel, I pack them. If it is a small parcel and I pack it very quickly. If it's a big one and if it's a fragile one, I take a lot of care in packing, right? And depending on which location it has to go, I have my pin code. So depending on the pin code, I know. But how do I know this protein has to go outside? Something has to be marked. You have to label that protein and keep, right? So that labeling happens in terms of chemical modification. If it is chemically modified by glycosidic bonds, then it has to go outside. Something, it is like a recognition. So that your degrading proteins do not come and destroy them. So in a cell you have degrading proteins as well, right? So your degrading proteins should not come and destroy something just like that. If there is a tag, it won't destroy. So if a sweeper is coming to sweep and if she finds a letter, okay, she will know it has an address, so it has to go there. So similarly, it is chemically modified just like you attach a pin code in your parcel. Okay? So this is glycosidation. So Satyendra is giving a lot of business to Amazon. Oh my God, what are you ordering? Order for us also. No, please order a cake. Please order some food for us also. Huh? So it's good if you're ordering books and studying. So Amazon is a very good platform. It serves a lot of people. It serves a lot of purpose. So I'm not denying. Okay, so this is your Golgi apparatus. Fine. So the next member is your lysosome. So here we are talking about your lysosome. They are membrane bound vesicular structures, but they have only a single membrane. So if this is your lysosome, they only have a single membrane like this. They are formed by the packaging in the Golgi complex. So sometimes some proteins that are packaged, they become a separate vesicle, that is your lysosomes. So yes, some proteins are packed. Sometimes when the proteins are packed, they become your lysosomes. But at Amazon, you don't get cakes, right? They don't deliver birthday cakes. That is your Swiggy. I'm sorry. So system of GERL, Golgi associated endoplasmic reticulum. So endoplasmic reticulum produces protein, your Golgi complex packs them. So when they pack, sometimes certain vesicles detach, that is your Golgi complex. So say if I am packing 100 cakes for delivery. But one cake, I am so tempted that I take it for myself. So it is something like this. So this becomes a separate membrane bound organelle, that is lysosomes. So some organelles, okay. So some organelles from your Golgi body, after getting packed, it moves out as your lysosomes. So system of GERL, Golgi associated endoplasmic reticulum. So rich in hydrolytic enzymes active at acidic pH. So what is present inside this? They are rich in hydrolytic enzymes. Enzymes are nothing but proteins. So your endoplasmic reticulum synthesizes them. Your Golgi body plaques them. The one that is having hydrolytic enzyme come out as your lysosomes. They are active at your acidic pH. They are polymorphic organelle. Poly is many. Morphic is types, the outside appearance, right? Morphology, morphic. So they have many external appearance. They do not have a specific shape. I cannot tell you lysosomes are spherical. They can take up any shape. Okay. So you have different orders in which your lysosomes are produced. Primary lysosome, then they become secondary lysosome, residual body, and autophagic lysosome. So pay attention. So primary lysosome, so I have my endoplasmic reticulum. So this produces proteins and goes to the Golgi complex. So this packs my protein. From the some packaging moves out as lysosomes. So what does lysosome contain? Hydrolytic enzymes, right? So these vesicles, when they are moving around the cell, so this lysosome which comes out from the Golgi complex and contains your hydrolytic enzyme, so this is known as your primary lysozyme. So the lysozyme that comes out of the Golgi complex, okay, containing your hydrolytic enzyme, that is your primary lysosome. Not lysozyme, lysosome. Lysozyme is an enzyme, lysosome is an organelle, right? So this lysozyme, this moves around the cell. So sometimes the lysosome fuses with a phagocytic cell. So phagocytic cell is the cell which engulfs food material. So your primary lysosome, it encounters a phagocytic cell and it fuses with that cell. So this forms my secondary lysosome. So your lysosome contains hydrolytic enzymes, so it acts on the food particles and it digests the food particles. So after digestion, whatever is remaining, that is known as your residual body. Okay. So now this residual body is not needed by the cell, right? So it undergoes cell death. It dies or it gets destroyed. So that is autophagic lysosome. So these are the four order in which happens. So that is why we say it is polymorphic. It does not stay in one stage. So did you understand what is primary lysosome? Which function? So phagocytic cell engulfs food material just like your endocytosis. So your lysosome contains enzymes, so they help in the digestion of food. So your food is digested, so the nutrients are absorbed. So whatever is undigested, that is your residual body, and further they undergo destruction. Yes, so Shimpi, did you understand? Did you understand lysosomes? Is it clear? Yes, Shimpi, did you understand? No doubt? Clear? So this is about your lysosome. So it has only one layer and it consists of hydrolytic enzyme. So Falguni, so I guess you're understanding. If you're not comfortable, let me know. So what are the enzymes that are present inside your lysosomes? So your nuclease, which acts on your nucleic acid. So an enzyme needs a substrate, right? For example, if I say A gives me C, so there should be an enzyme. So this is your substrate and this is your product. So your enzyme acts on the substrate to give your product. So your substrate is nucleic acid, the enzyme is nuclease, okay? So please repeat the 4 parts, alright. So from your endoplasmic reticulum, your proteins are produced, they move into the Golgi complex. So your Golgi complex parts, among them some move out as separate vesicles known as lysosomes. They contain your hydrolytic enzyme. This is your primary lysosome. Okay, so sometimes your primary lysosome encounters phagocytic cell. So this contains enzymes, this contains food particles. So they fuse with each other, that is your secondary lysozyme, right? So now the enzymes act upon the food and they digest them. So whatever is digested is observed as nutrients. So whatever is undigested is known as your residual body. Now your residual body has no function, so it undergoes death, it undergoes destruction, that is your autophagic lysosome. Okay, Antonia B? Yes? Clear? You have understood? Should I repeat it once again? No? Can I move forward? Yes, can I move forward now? Okay. So protease acts on protein, glycosidase acts on your polysaccharides, your lipase acts on your lipids, your phosphatase acts on your phosphate linked compounds, sulfate acts on your sulfate linked compounds. Done? So this is about the enzymes in your lysozymes. So the last member of your endomembrane system are your vacuoles. They are membrane bound space which are present in the cytoplasm. They contain water, sap, meaning some amount of solutes or ion, excretory product, and the products which are not useful for the cell. So they have a membrane, they are membrane bound. The membrane of your vacuole is known as tonoplast. So vacuoles occupy 90% of the cell volume, especially in case of your plant cell. Done? So example, you have contractile vacuole that helps in osmoregulation, maintaining the osmotic concentration of the cell in amoeba. So Ashley, ma'am, is it possible to get 650 if I start now? So are you going to revise now or are you going to start from scratch right now? Huh, Ashley? Okay, where do you get the PDF? Yes. So please download eCareerPoint app. So the symbol, so it is like this, eCareerPoint app from your Play Store. So once you download it from your Google Play Store, so please log in with your Google ID. So if you log in, so then if you open it, so in the panel below you have free stuff. So in the free stuff you will get all the YouTube PPTs, but the name of our channel is eCareerPoint-English medium channel. So go to that and get the biology PDFs, okay? So that is how, so you get it in eCareerPoint NEET channel. So you have to go to the down panel and click on free stuff, right? Is it clear? So Ashley says 50% syllabus is completed. Okay, if 50% is completed, yes, then if you put on more hard work, if you study for more than 5-6 hours a day, yes, it is really possible, if provided if the 50% you are very strong. Provided if the 50% you are very strong, and yes, you can really get ahead. Okay, Ashley? So a contractile vacuole is example, so they help in the osmoregulation in amoeba. So for example, protistans, which are unicellular eukaryotes, the food vacuoles are formed by engulfing food particles. So this is about your vacuoles. So the membrane is your tonoplast, they are the central vacuole, they have cell sap. So this is about your vacuoles, right? Yes, so with this I stop. So I am done with my endomembrane system. So in the next class I will bring some questions and then move forward with the next topic. So once again I have a reminder. So tomorrow at 4 p.m. so we are meeting talking about the respiration in plants, and then at 6:30 we have a surprise mentee. So it is episode 4. So please join me for the same. So if you genuinely liked my way of teaching, so please do like, share and subscribe and join me every day. So standard 4 to 5 we are having class every day. So please meet me in the same. So until then, stay safe. So take care. I'm waiting to see you tomorrow. So bye and take care.